So now we would like to invite both Natalie and Francesca for the stage for the discussion. And the floor is open for discussion, so you need to raise your hand when you have a question and then wait for the microphone. So then it will be with you in a bit. And I also encourage people online to put in some questions in the chat. Yes, is this working, the microphone? No, it's not working? Can we get
Try without. Try without.
But for the online speakers.
For online people, yeah. Sorry, Renato. We need the microphone because you're online.
Yes.
Say your name, please, and country.
Yes, Renato Fenchin from France. I would like to congratulate the two speakers with this very interesting presentations and complementary presentations. I would like to ask a question to Francesca. Of course, Natalie's data, we are more familiar with it, but the role of LH that you are proposing now is, for me, a little bit puzzling. And I would like to know if you consider that we could use other agents in the same model that used for LH to show if you had the same biological effect, like maybe FSH or androgens. If you could have the same results, even more results. I don't know if you consider doing this kind of experiment.
Is it working?
No. Oh, yes, please.
Yes, so initially when we started analyzing the effect of LH as a chemoprotective adjuvant, we tested also FSH. And we did see some protection, let's say, but not as strong as LH. That's why then we kept going on with LH and not anymore FSH. We tested others. We tested we tested testosterone at one point as well, and we had no effect with that. Then we left all the type of hormones and we went to see other molecules more specific in the pathways for to protect the ovaries and the apoptotic pathways. That's what we did. In terms now of this new, let's say, this new project, this new effect that we are seeing, no, we are focusing at the moment only on LH because that's what we would like to see. That's an exchange. I can come over here.
But the data that you obtained with FSH and androgens you didn't publish?
No. It's a supplementary figure of the first paper by Orsi in 2017. Yes, FSH, we have that, but then that's all.
And the last short question, what was your rationale for this unexpected action of LH?
Our idea was that normally, actually, with the chemotherapeutic drugs and the antagonists that normally is given to patient, you put in quiescent the ovary. So actually, it should be exactly the opposite of what we're doing. But anyway, you see an initial peak of LH and then the down regulation. So Our idea is that we are giving in a very, very short time. It's just really 24 hours, and then we see the rescue. So it's it's a way to activate a pathway, probably. So it's LH, but LH is the activator of something else that we still are investigating, and that was our rationale.
Thank you. Francesca, a question for you. So you showed that in, I think in mice, LH may increase ovarian volume. So very easy to understand how can LH act onto follicles. But how can LH increase ovarian volume?
Well, increases the volume because it increases the volume of the follicles.
Okay,
That's
because of
what
that.
so we measured what we first did was the normal histology, so sections, and then the counting, and we had the idea looking that the volume was bigger, but we couldn't, it was not very precise, just measuring
So
with
can you exclude
centimeters.
the possible effect of LH on ovarian stroma? I mean, if I go to the clinical side of the story, you may agree women enter menopause because they finish the ovarian reserve, the follicles are no more there. But if you calculate the ovarian volume by ultrasound at 52 years of age and at 80 years of age, you will find a different volume. I mean, the volume of the ovary is going down across female, with female aging. So, I mean, can we exclude that gonadotrophins may act on ovarian stroma?
We do not see, no, we do not see any increase, let's say, in the stroma. We haven't performed specifically then analysis on the stroma. What we thought was at some point collagen. We tried to measure the amount of collagen if that was changing. But from what we see now, no, there's no change exactly in the stroma.
Question in the back first.
This is the microphone.
I think it might be working. I think it might be working. Let's see.
It's working now.
Let's see.
Thank you so much for two very, very brilliant talks. I have the first question applies to both talks because I'm curious about physiological role for both AMH and LH since in the fetal ovary in very early stages of follicle development because during fetal ovarian growth there shouldn't be LH, the ligand shouldn't be available. So what's the reason for expressing the receptor? Does it play a physiological role? Same thing for AMH, because AMH appears very nice functional data, very nice review. It appears to effect activation in early, but we know that AMH is produced only by granulosa cells from growing follicles. So what would be the role? And lastly, Francesca, if you could help us to understand the effects that we've just published on follicle quality, on oocyte quality. We've just published that the LH priming can double pregnancy rates in Poseidon 4 patients. And we surprisingly saw an effect on oocyte quality. Oocyte numbers only in those patients very, very, in a very bad situation with very low levels of AMH. So could you help us to understand the impact on oocyte quality?
Okay. Thank you. Yes, you are right. The known expression profile of AMH indicates that AMH starts to be expressed by primary follicles. And it is true also that according to the work of Paolo Giacobini, AMH doesn't cross the placenta barrier. But again, I don't think that, I think that we should reanalyze the expression of AMH during the late stages of gestation or pregnancy. Because as you know, the results depends upon the antibodies you are going to use. And perhaps there are, and you know, there are different forms also of AMH. This is something very, very difficult to understand up to now, as well as the expression of the AMH type 2 receptors very early in the fetal ovaries.
So for instead LH, there is a paper published by others that they did find already the receptor even in fetal gonads. Okay, so it's a period that you're not even expecting it at all. What is then there the physiological role? I do not know. What we can say is that we have analyzed a situation that is not physiological. We're giving the chemotherapy, so probably there is a damage, we are doing something, and then LH can promote, activate pathways, okay? So that will not be a physiological way of using it. The other is now in this idea of being a promoter, again, we are giving anyway a high amount of LH in this case. High for mice, I would not exactly know then what will be the use of the amount of LH for a patient, because also I think receptors, the expression of the receptor might vary in these stages, so we need to for sure do a more accurate analysis of all this. On terms of the quality of the oocyte, what we have seen so is when we give the damage, so we have to induce a damage then to observe the effect of LH. We have the damage of the DNA, okay? So those oocytes are definitely damaged, but and then that causes death they activate PA63 and that activates apoptosis, and so we lose these oocytes. When we give LH, we have observed that damage is there, but then tends to be repaired. The marker that we are using is gamma H2AX, so when you do see a decrease in gamma H2AX, you have it's said that you have a decrease in the damage on the DNA. So at the moment, that's our idea of how it's working. But we tried, and it's a very initial project, to analyze all the repair genes and all the mechanism that should be underneath this mechanism, but I do not know yet. I hope to answer you in the future. Okay.
Okay, more from the floor? Oh, Olivia?
Thank you. So I would have a question for both of you. The first for you, Francesca, but yeah, your data are very nice because basically suggest that the LH receptor is expressed in preantral follicle, so it in a certain way debunks the classical concept from endocrinology by which those receptors are instead expressed only in the antral stage. So admit that you will demonstrate and consolidate the data even in human. So what do you expect is the role of the monthly LH surge on the whole follicle set of the ovary? And also, if you may hypothesize that maybe they could be a reminiscence of the fetal stage is when the ACG may act on those receptors to push the development of the gonads through the production of steroids. So in that case, if it is true, maybe LH may do not act on those follicles, but ACG instead, yes. So what is your opinion in this context, if I have explained that?
That's a...
Yes, in the mice, of course, it's not possible because the mice has not a chorionic gonadotropin, but in human maybe. And I take the occasion to have the microphone to set the question for you, Natalie. Thank you for your talk, also very nice. Maybe you told this, but I missed something. Are known some anti-Mullerian hormone receptor mutation and their effect in the antral stage or preantral stage?
Oh. There are the mutation of the AMH and AMH2 genes lead to in males to a pathology called persistent Mullerian-Dux syndrome. And it's true that it was a pity, but we didn't succeed to analyze the sisters the sisters of all these children. So we cannot say a lot of things about these women with a mutation probably of these genes. Jenny Fischer performed several experiments on mutation or at least variants, and to analyze the effects again of AMH on folliculogenesis. It was a way to analyze that, and they showed that some variants were associated with lower estradiol levels, and because in general these were for variants often associated with PCOS, for example, and on the contrary, they suggest that some variants could be related to ovarian failure. But
wonderful. So Natalie, so how do you? Okay, so in the antral follicles, AMH reduce aromatase expression and estradiol.
yes.
And how do you see that in the context of PCOS? Because there we have very high AMH. What comes first? What is regulating what?
Okay, so I would say that in the PCOS, AMH has various effects. Of course, for me, it inhibits follicle differentiation, leading to a follicular arrest and the absence of selection of a dominant follicle. But also AMH contributes to increase the follicle pool because of its positive effect on androgen production. And since it is going to increase androgen production, of course, it is perhaps going also to via androgens, to increase the primordial follicle activation.
So what do you think comes first? Is it the androgens affecting the follicles to grow more and produce more AMH, or is there a higher production of AMH within the granulosa cells, and then that creates that?
It's hard to say.
I know.
It's hard to say. we are carrying out studies to, in order perhaps to answer to this question, we are carrying out studies on a model of PCOS, which is called the Goto-Kakizaki rats, and we try to analyze the ontogeny of the appearance of the phenotype. And it seems that this phenotype could arise from disturbances very early in the ovarian development.
Yeah, I think you're right. So I think in our studies, we have also looked into the content of AMH in these follicle fluid in small antral, and normally at follicular selection, we see AMH production goes down, and in PCOS, it stays high.
Yeah.
So it's definitely something about the whole population of AMH. And
yes, it's true. We have also shown that in PCOS granular cells, luteinized granular cells. It's why I didn't talk about that today, because we focus more today on preantral follicles, and but we have shown that a lot of factors can upregulate both AMH and AMH receptor, androgens, LH, estradiol.
And what do you think of the oocyte-specific factors, the TGF9?
Yes, BMP15 and GF9. In the PCOS context, you mean? I have no idea.
No, I don't either.
Because, as you know, it's very difficult to measure the levels of BMPs in the blood serum. I think that BMPs, a bit like AMH, not in the context of the PCOS, but they promote follicle growth and inhibit follicle differentiation. Yeah. But in the context of PCOS...
Nathalie, again, on AMH, I mean, we know that, at least up to now, we know that the only endocrinological action of AMH is to lead to Mullerian duct regression.
Yes,
okay?
yes, right.
So the role of AMH is still a paracrine role. AMH has a relevant inhibitory effect on cells. It's a signal of cell death, at least onto Mullerian tissue. Yeah. And on the ovary, you showed it may have different effect. It may be a survival factor on atretic follicles, but then it may be again inhibitory on primordial follicles. But we are talking here about granulosa cells. So how can you explain this different effect of AMH on the same type of cells? And the second question is, we know that AMH in some way has a relevant role onto ovarian function, and recombinant AMH has been developed for research purposes many years ago, probably 15 years ago. And I know for sure that some startup companies or some pharma companies have tried in some way to develop something for human use. Why recombinant AMH is still not with us?
I don't know. I don't know for the last question, I mean. I think that we can purify very properly AMH, so it could be tried for clinic. I think, but I don't know why the companies does not want to invest money in that. I cannot answer to this question. Regarding the effects of AMH on granular cell apoptosis, It's true that, again, there are discrepancies upon the studies, but I think that, like a lot of factors, AMH might have different effects depending upon the stage of differentiation of the granulosa cells. But it's hard to confirm that. It's true, you're right. But again, I have not the answer, and I just I can only try to see among the literature the number of articles in keeping with an effect or not. But no, I have not the molecular answer to this question. We have identified some genes which are regulated by AMH and which are involved in granulosa cell death or survival, but we didn't have the time to study more these genes.
Okay. So Francesca, you didn't show your data on the inflammatory studies, but could you please still comment? Because I think it's an area that we haven't researched a lot, the whole immunology and ORI and inflammatory effects.
So first I needed to reply to Livio, not to Livio, no, because I've been thinking, I had time to think to the answer. So we are in two different situations. Our studies are two different situations. One, again, is damage. The other one should be a physiological, but again, it's not 100% physiological because we are stimulating something with LH anyway, and we haven't measured the amount of LH that is present in these PAPs in the blood. In relation to the damage that we give is, as I was saying, very short treatment, okay? On the other side, instead, is a sort of prolonged treatment. So I guess that, I mean, in the normal cycle that you were saying, so we get the peak, we are like always not at the level of the peak, but anyway, we are at a high level, okay, of LH constantly. So that's why I think in a normal cycle, those primordial follicles might sense the LH, but will be reacting more, just being healthier, okay, and not going in the activation. So what we think is, if there's a damage, LH promotes anti-apoptotic pathway. If we are at a low level of LH, and the receptor is there, and so those follicles can reply to LH, they are healthier, but they stay better, but questioned anyway. If we stimulate with a higher dose of a constant dose of LH, then they might promote an activation. This is a general idea of what it thinks. And this so is the pro-inflammatory. We went to check some interleukins. We did not go through the whole cards that are available. We went just for some interleukins, and we did see that they were upregulated with the chemotherapy, and instead downregulated with LH. It was interleukin 6, if I do not remember wrong, and interleukin 3, that were the two that we analyzed. We would like to do the whole panel that is available, but we had to concentrate on.
We have one question from the online audience. Yes, okay. Again, on AMH and men, what's the role of AMH in the testis, and is there any reason why should we measure AMH in infertile men? Good question. Good question.
Okay, first question. We used to work on the effects of AMH on the testes, because here again, AMH is expressed in postnatal children. And the levels of AMH are very high compared to women. So we wanted to analyze the effects of AMH, and we showed, and our team also confirmed and also showed this result, that AMH can repress steroidogenesis. Leydig cell, of course, steroidogenesis. And in both the fetal population of Leydig cells and the adult population of Leydig cells. And we also, several teams showed that AMH could also repress aromatase in Sertoli cells.
Yeah.
So these are, of course, in vitro studies, basic research studies, and it's how to say to understand the relevance of these results in human.
Professoressa Simoni?
Manuela Simoni from Modena, here, around the corner. In my opinion, AMH in the adult, so the function in the male, we know it's a regression of Mullerian ducts. In the adult, both in female and in males, is a paracrine factor. So it is produced locally, and it does something locally in the ovary and in the testes that we don't really know yet what it is. When we talk about AMH in the adult, we have to stop to think about endocrinology, because this is the reason, Antonio, why it did not have any use as a drug, because you give a drug and the drug goes everywhere. So if the effect is paracrine, then I think we should perform some in vitro experiments, because paracrinology in vivo is impossible so far. And we should use new technologies. For example, single-cell RNA-seq could give some answer. So it's really something that we have to change in our mind. We should think about studying the paracrinological effect of these factors with new methodologies and using also all the informatics behind it. And to answer the question of the person online, I don't think there is any clinical role of measurement of LH in the male at the moment, in our experience.
Okay. I can remember and recall just one article in which the authors treated a hypo man with a recombinant FSH, and they measured the AMH as a way to test the testicular response to gonadotrophins. But that's the only paper to
Yes, it is true that AMH is FSH dependent. So if you give FSH, then you increase MH levels.
Yeah.
But what is it doing? Nobody knows.
Sure.
At the moment, for us clinically, AMH in the male is a marker of the presence of the testicle.
Yeah.
So if you have a cryptorchid boy and you measure MH, if you measure MH, then the testis is somewhere.
And the marker of puberty also.
Yeah. You're right. I mean, sometimes we have intersex conditions in baby born, in newborns, and you can measure AMH just to understand whether that's a man or it's a male or female. Please, José, last question.
Yes, thank you. I forgot to introduce myself. I'm José Burattini from Eugene, Italy, and São Paulo State University. Just a comment on the paracrine and also to stimulate the discussion. It was mentioned very precisely that AMH decreases cyclic AMP activation, but also the expression of the FSH receptor. And perhaps it's a valid hypothesis to consider AMH as a modulator, because we wouldn't want to empty the ovarian reserve out of a sudden, so it could be at that time a modulator. And at a later stage, you have just published that cumulus cells express AMH, and the oocyte increases the expression of AMH while reducing the expression of the FSH receptor, as if it was trying to prevent excessive FSH activity around itself. So I think just some clues, some comments on the paracrine. I fully agree with the paracrine importance and role of AMH.
Yes, then you know that some authors showed that AMH was able to regulate the GnRH neurons. So even if there is a local production of AMH by GnRH neurons, most of AMH is produced, of course, by the ovaries. So this suggests perhaps an endocrine role of AMH to the brain.
Yeah. Okay, so thank you for this, and for young researchers and students and doctors here, you should know that in 2007, many years ago, we held here in Modena the first Congress on AMH. It was a national campus, AMH, what do we still need to know? 17 years later, we still need to know many things. But I was thinking probably it's time to have another international Congress on AMH, maybe next year here in Modena. So an AMH reloaded, yeah, could be. Okay, so coffee time now, see you in 20 minutes.